So now that we've collected plenty of electrons from all of the other phases of cellular respiration, NAD and FAD carrying their electrons and protons in the form of NADH and FADH2 will come to the electron transport chain to drop off their electrons and transfer their energy into ATP. The electron transport chain is a series of membrane-bound electron carriers. They're in the inner mitochondrial membrane. So NADH comes from the mitochondrial matrix, the fluid inside the mitochondria, and drops off electrons at the first electron acceptor in the chain. Those electrons will then be passed from electron acceptor to electron acceptor all the way down the electron transport chain until they're transferred to oxygen by the final membrane-embedded electron acceptor. Essentially, as they bounce down through this stairway of embedded proteins, the electrons lose energy. As they lose energy, hydrogen ions are pumped through these electron acceptors into the intermembrane space. As the electrons pass each embedded protein, they pump more electrons into the intermembrane space. So the density of protons in the intermembrane space is becoming very thick. These proton pumps that are also electron acceptors are creating a proton gradient as they transfer electrons to the next carrier. Again, the final electron acceptor is 1/2 of an O2. And we take two hydrogens and put them together with 1/2 of an O2. We have water. So this is where water is generated and oxygen is consumed in the process of cellular respiration. You might recall that both of those are part of our initial equation. Be sure that you look into that initial equation and understand where the carbons go and where water comes from and where the carbon dioxide comes from. It's important stuff. So let's take a quick look at the analogy. What's going on in here in the prom analogy? What we have is the NADH limos coming along and dropping off their electrons. Remember, we said the electrons are girls and the hydrogens are boys. When they show up at the door guy is kind of a rotten jock sort of guy, and he wants to take all the girls for himself. And so he says, "Welcome on in, ladies. Come on down the stairs." And the ladies bounce down the stairs all the way down until they enter prom, where they're received by this really nice guy, Oxygen. Oxygen says, "Don't worry, ladies. We'll get your guys back. I'm going to take care of you until they get here." The meantime, the guy at the door has not permitted the guys to go in. Remember, the guys are hydrogens. He's thrown them out literally in the back alley. He's that kind of guy. So as they try to go down the stairs, these bouncer guys keep throwing them out into the back alley. And naturally, they're pissed. So they're running around in the back alley, lots and lots and lots of hydrogens, and they really, really want to get into prom. They are pissed off. So how are they going to get back in? They're running around in the back alley looking for an entrance to get back in. And as it turns out, there's a revolving door. Hydrogens find this doorway and they rush in through the revolving door, spinning it so fast that it makes ATP as they're on their way in. And then the hydrogens, wow, look at that. The hydrogens are the ones that come back here and join up with 1/2 O2 and the electrons. And now we have a very happy water molecule. The couple is back together. So kind of a goofy story, I know. So let's look in more detail at this revolving back door. This is the money. This is where all the ATP is made. This is the key to the whole thing. So the majority of ATP is made during the electron transport chain and chemiosmosis because of this proton gradient. So you'll recall that the membrane proteins have been pumping hydrogen ions across as they accept electrons and move them down into the main event where they're received by oxygen. This accumulation of protons in the intermembrane space drives the protons into the matrix via diffusion. They really want to move down their concentration gradient. And there's such a great pressure, such a great drive, they literally spin this molecular motor. The molecular motor spins, and as it spins, the energy of the hydrogens pushing through spin it so fast that it catalyzes the phosphorylation of ADP to form ATP. Now, the inner mitochondrial membrane is relatively impermeable to ions, so most of them move through this bump. However, some of them will re-enter the matrix through the spaces in the membrane. So the membrane is a little bit leaky, but most of them go through this revolving door, which is called ATP synthase. So what about the FADH2s? Well, the FADH2s drop off their electrons at a slightly lower energy level. They drop them off at the second electron acceptor in the electron transport chain. And then they bounce down the stairway of proteins in the same fashion that the ones did from NADH. However, only 2 protons are pumped for each electron that's transported, so there's a slightly lower energy yield for FADH2's electrons and protons than there is for NADH. I think of it as the lower budget limousine. So again, to review, electrons are dropped off in the electron transport chain. As they bounce down the energy levels of the electron acceptors in the electron transport chain, much like a stairway, this energy fuels the pumping of hydrogens into the intermembrane space. The electrons are accepted by 1/2 of an O2. Now we call it 1/2 of an O2 because an oxygen simply won't exist on its own. It's way too electronegative. It needs to be stuck to something. But for just a moment, 1/2 of an O2 or one oxygen is free and it can pick up the electrons and the hydrogens that have zipped through this revolving door, creating all of the ATP. So let's see about diagramming the electron transport chain and chemiosmosis. This one's a little bit tougher. Basically, I've drawn 2 different membranes. We have the inner mitochondrial membrane and the outer mitochondrial membrane. Again, hopefully yours will look more neat than mine. Here I've drawn the electron transport chain to represent stairs and ATP synthase, where we'll see the majority of ATP produced. So NADHs will come in to drop off their electrons at the electron transport chain. Here we're seeing that NAD is recycled. It can now go back up to glycolysis and pick up electrons, or to the Krebs cycle and pick up electrons. But this is the recycle spot for those. The electrons will move in pairs, and they will bounce down the electron chain until they get to the final electron acceptor. The final electron acceptor is 1/2 of an O2. As the electrons are bumped down the electron transport chain, hydrogen ions are transported into the intermembrane space by each of the membrane embedded components of the electron transport chain. So we're getting lots and lots and lots of hydrogens stuck in this intermembrane space. We have a strong hydrogen gradient, and they really want to move down their gradient and get back in to the mitochondrial matrix, which is over here. So hydrogen ions have now found the back door, the revolving door, and so they will race through the ATP synthase, spinning the door as they come through. They're moving so fast that ATP synthase is able to phosphorylate the formation of ADP into ATP. This is where most of the ATP is made in cellular respiration. Now that we have these hydrogen ions, they'll come back and meet up with the 1/2 O2, again in a pair. And when we have hydrogen ions and 1/2 O2 coming together, we end up with the formation of H2O. This is where all the water is made in cellular respiration. So again, this would be a good rough draft of your diagram, but you'd want to neaten it up in order to pass it in for your diagramming assignment. A quick animation of the electron transport chain and chemiosmosis.
The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
So now we have all 4 phases of cellular respiration in the aerobic condition covered. Glycolysis, which happens out in the cytosol of the cell, produces pyruvate. The pyruvate is then oxidized inside the matrix of the mitochondria. Krebs cycle also occurs in the matrix of mitochondria. Each of these processes produce a little bit of ATP, but a lot of NADH and some FADH. These are the electron carriers that carry electrons to the electron transport chain. Once at the electron transport chain, the energy of electrons is captured to pump hydrogen ions out into the intermembrane space. Once there's a hydrogen ion gradient here, the hydrogen ions will want to rush back through ATP synthase. As they do, they turn a little molecular motor. This molecular motor is enough to power the production of ATP from ADP and inorganic phosphate. As the hydrogen ions come back through the ATP synthase into the matrix of the mitochondria, oxygen is split, picking up the electrons from the electron transport chain, and the hydrogens come together and they form water. So that's everything in a nutshell. Seems pretty simple, right? It does take a lot of practice to understand how all of these components come together. So please watch all the animations out there and spend some time working on your diagrams. So let's look at the energy yields of respiration. Theoretically, we have 38 ATP for each glucose in bacteria and 36 ATP per glucose in eukaryotes. The actual energy yield, though, is more like 30 ATP per glucose for eukaryotes. And this reduced yield is because that inner mitochondrial membrane has a little bit of a leak for hydrogen ions. That is, not every single hydrogen ion goes through the ATP synthase. There's such a strong gradient that some of it sneaks in between those phospholipids. In addition, the proton gradient could be used for purposes other than ATP synthesis, like co-transport of another molecule that needs to move up its gradient while hydrogen ions are moving down the gradient. This figure is a wonderful summary figure to help you understand where all of the different ATP comes from. One thing that's important to notice is that very little ATP is generated in glycolysis, pyruvate oxidation, and the Krebs cycle. The majority of the ATP is generated by chemiosmosis after the electron transport chain. What we do get from each of these three stages is lots of electron carriers. And remember, electrons represent energy. As we pull electrons away, carbons are freed in the form of carbon dioxide. And all the NADH that's collected runs to the electron transport chain where we can generate lots of ATP. Now, how is all of this regulated? What if there's enough ATP already and we don't need to make any ATP? Well, that happens to be one of the key control points. In glycolysis, there's a keystone enzyme called phosphofructokinase. And if there's a lot of ATP production, the ATP itself feeds back to shut down the action of phosphofructokinase so that the glycolysis reactions no longer proceed. Now, if there's a lot of ADP, ADP actually allosterically activates phosphofructokinase so that we can turn on the system of glycolysis in order to make ATP. So this is a great example of what we looked at a couple of chapters ago of allosteric activation and allosteric inhibition. The ATP and the ADP bind to something other than the active site of phosphofructokinase. Phosphofructokinase's active site is specific for fructose 6-phosphate. Another place that we regulate ATP production is in pyruvate oxidation. The enzyme pyruvate dehydrogenase is inhibited when there are high levels of NADH. If we have high levels of NADH, then we're well on our way to producing plenty of ATP. Another enzyme is citrate synthetase. It's inhibited by high levels of ATP. We find this enzyme in the Krebs cycle. Now, what about if oxygen is not present? We've spent a lot of time looking at oxygen being present. If oxygen's not present, we're going to be undergoing only the glycolysis portion and some anaerobic respiration. In anaerobic respiration, you'll recall we use inorganic molecules, something other than O2, as a final electron acceptor. Many prokaryotes use things like sulfur or nitrate, carbon dioxide, or even inorganic metals. In fermentation, we're using an organic molecule as the final electron acceptor. So let's take a closer look. Methanogens are anaerobic bacteria. In methanogens, carbon dioxide is the final electron acceptor, and it becomes reduced to form methane, or CH4. It's the methanogens in the cow's gut that can break down those beta-glycosidic linkages between beta-glucose, and in doing so, they produce a lot of methane. Sulfur bacteria use inorganic sulfate as the final electron acceptor, and it's reduced to hydrogen sulfide. This is where we get all the sulfur in these sulfur hot springs. These are the early bacteria that set the stage for evolution of photosynthesis itself. We'll discuss that further in another chapter. In fermentation, organic molecules are reduced in order to regenerate the NAD to keep glycolysis going. In ethanol fermentation, we see occurring in yeast. CO2, ethanol, and NAD are produced. Whereas in the lactic acid fermentation that we see in our cells and other animal cells, especially in the muscles, the electrons are transferred from NADH to pyruvate to produce lactic acid. Of course, that lactic acid buildup can only build up so much, and then we have to return the lactate to pyruvate and put it through the oxidative processes of cellular respiration. Now, how do things other than carbohydrates get fed into the cellular respiration scheme? Well, amino acids have to first undergo a process called deamination. That basically removes the amino group. The remainder of the amino acid is converted into a molecule that can either enter glycolysis or the Krebs cycle. For example, alanine is converted directly into pyruvate, so it can enter right where pyruvate is. And aspartate is converted to oxaloacetate, and so its carbons can enter in the Krebs cycle. Fat is broken down into fatty acids and glycerol. Those fatty acids are going to be converted into 2-carbon acetyl groups. Basically, we chop off the carbons 2 at a time in a process called beta oxidation. This is an oxygen-dependent process, and thus fats have to be burned in the presence of oxygen. That'll explain why only aerobic metabolism will burn fats. The respiration of a 6-carbon fatty acid yields 20% more energy than a 6-carbon glucose molecule. This is why fats are such a rich fuel storage supply. This figure summarizes how many of the molecules that we consume filter into the cellular respiration process. You don't need to memorize it by any means. It's just interesting to note where the different macromolecules come in to be digested so that they could be broken down and their energy can be converted into ATP. Let's take a quick card quiz. Most of the ATP that's made by aerobic respiration is made during which stage? The process of removing an amino group from amino acid is beta oxidation, is used to extract energy from. What happens to the electron's energy as it moves through the electron transport chain? Here are your answers. So now, your duty is to make sure that you get a full diagram of cellular respiration, including some fermentation to both lactic acid as well as to ethanol. Please take the time to make sure that these diagrams are clear for you and contact me if you have any questions.